Okay, so we can start. Hello and welcome everyone to the live session of week 12 on the course Experimental Biochemistry, which has been taken by Professor Shagata Das Gupta and Professor Shomode of IIT Kharagpur. I am Srijan Sen, and today I will be discussing about the lectures which were taught to you in the week 12, and also discussing with you a few of the assignments and questions which will help you in solving your assignments and taking up the examination which is coming up. Okay, so I am sharing my slide. Am I audible, and is my screen visible? Am I audible properly, because there is actually a Wi-Fi connection problem. So am I audible properly? Okay, that's good. Okay, so I think we can start. So this is basically the last live session class I will be taking, and on Sunday I have kept just a small quiz, or you can say a summary session, where I will be discussing only questions. There won't be any lectures, only a few questions. So you people are welcome to actually come and attend those quizzes and lectures. So it's a summary of all the lectures which has been taken. So that is going to be a 1 hour class on Sunday from 4 to 5. So all are welcome, and you can also tell your friends if they want to join the class. It's going to be a 1 hour class. So today is officially the last week live session which I am taking for this um, January to April course 2023. So in today's live session, we will be discussing mostly about blotting, the different types of blotting and all. Okay. So what is blotting? So, sorry. So, blotting is a technique for you can say transferring DNA or RNA or the other nucleic acids and proteins onto a carrier so that they can be separated, and often follows the use of a gel electrophoresis. So basically the southern blot is used for transferring of DNA, the northern blot for RNA, and the western blot is for protein. So what we do over here, we actually transfer the different types of biological molecules, which includes your DNA or RNA or sometimes your proteins, onto a carrier where we would be visualizing. So this carrier is important, where we would be visualizing. And so primarily what we do is that we separate them using any kind of gel electrophoresis, and after that we transfer it over some kind of media, and then that carrier thing is used for visualization. So there are basically 4 blotting techniques. One is the eastern blot, we have the western blot, the southern blot, and the northern blot. So we know that northern blot is for detecting of RNA, the southern blot for the detecting of DNA, western blot we perform for detection of proteins, and eastern blot is for detection of protein post-translational modifications. So this is a very rare type of blot which we do for understanding the PTMs, or the post-translational modifications. So moving on to the first technique, which is our southern blot, or the southern blotting technique. So this technique was first developed by E.M. Southern in the year of 1975. So we use this technique to detect the presence of particular pieces of DNA inside a sample. So the DNA can be detected, can be it a single gene, or it can be a larger piece of DNA such as a viral genome. So it can be the different things. So the DNA can be in the form of a single gene or a larger piece which is cleaved, and then we want to see it on a gel. So it is, but basically it is for the detection of the presence of DNA inside a sample. So what we do, the southern blot combines agarose gel electrophoresis for size separation of DNA with methods to transfer the size separated DNA to a filter membrane for probe hybridization. So this part is important. Okay, so it combines, first of all, is agarose gel electrophoresis for size separation. So first we separate it based on size using agarose gel electrophoresis, and then we transfer the size separated DNA to a filter membrane, and then comes the transfer for probe hybridization. Now what is this hybridization? So this is the process of forming your double-stranded DNA molecule. So we are forming a double-stranded DNA molecule between your single-stranded DNA probe and your single-stranded target patient DNA. Okay, so we are forming a double-stranded DNA between one which will act as a probe, and second the DNA which we are wanting to target. Okay, so this is what we are expecting to see from this southern blot. So, and this is what is hybridization. So what is the principle behind southern blotting? So the mixture of molecules is first separated, then the molecules are immobilized on a matrix. Now the probe is added to the matrix to bind the molecule. So then we add the probe to the matrix such that it binds with the molecules, and then we wash it to remove any kind of unbound probes. Now only those places where the probe gets connected, it corresponds to the location of your immobilized target molecule. So only those places where your probe gets attached, there we can actually find the target molecules, because only the probe is going to, it's specific and it's going to only it's made only to target our target molecule. So it's going to get attached to our target molecule, and then we can see the location of that particular molecule. Okay, so this is the apparatus. So what is this apparatus? This is where we perform the agarose gel electrophoresis, and in this place we do is the southern blotting. So this is the your schematic diagram of your the two apparatus. So what we do over here, so there from the bottom, if we see, we have so this is a capillary plotting apparatus. So we have a plastic tray over here, then we have the gel followed by the membrane. After that we have three sheets of three, sometimes we keep 5 or 6 depending on the we keep filter paper, and then we have some paper tissues, and above it we have the glass plate with a weight which is less than 750 grams. Okay, so this is the actual pattern in which we place our gel for the transfer process. Okay, so first of all we need to keep in mind is that just above the plastic tray we are going to place the agarose gel, and then we will be having the membrane where we are going to transfer it. So the gel is where we have separated them according to their size. Then we have the membrane. After that we have 3 sheets of filter paper, and then we have other layers, which includes the paper tissues and the glass plate, and this weight is given such that there is no disbalance and there is uniform transfer between the gel and the membrane. So what are the different steps that we see in southern blotting? So first we digest the DNA with an appropriate restriction enzyme. So we get the digested, we cut it into small pieces, and then we run it on a gel to separate them according to their size, and you see that the migration occurs, that the larger one lags behind and the smaller one runs faster. So then we separate them according to their sizes in the gel electrophoresis. Now the restriction fragments present in the gel are denatured with alkali and transferred onto a nitrocellulose filter or nylon membrane by blotting. So then we blot it on a nitrocellulose filter or a nylon membrane, and then what we do, this procedure preserves the distribution of fragments in the gel, creating a replica of the gel on the filter. So first we run the gel and then we place it on a so this is important as to what is the composition of the membrane that we are using. It's a nitrocellulose filter or a nylon membrane, and these are the DNA fragments which gets transferred from the gel. So in this pattern it's moving up and it's getting transferred on the membrane, and this actually preserves the distribution of fragments in the gel, creating a replica of the gel on the filter. Now what we do, the filter is incubated under hybridization conditions with the specific radiolabeled DNA probe. Now we radiolabel it with a particular DNA probe which is going to form a double-stranded DNA. Now this probe hybridizes to the complementary DNA restriction fragment, and after that what we do is the excess probe is washed away and the probe is bound to the filter, which we are going to detect by autoradiography, which reveals the DNA fragment to which the probe is hybridized. So once more I am repeating it. So first we digest it with a restriction enzyme, then we run it and separate it based on this size. After that we denature it with okay, so this step is also important, that we denature it with alkali and transfer it onto a nitrocellulose filter or a nylon membrane, and this helps to preserve it in and preserve the fragments the gel and create a replica of it on the filter, and then we use radio label DNA probe to hybridize it, and then the excess probe is washed away, and only the probe which binds to the filter is detected by your autoradiography, which reveals the DNA fragment to which the probe is hybridized. So this is the summary of the blotting method. So extract and purify DNA from the cells. DNA is restriction digested with enzymes, then we sort it with electrophoresis, then we denature the DNA using alkali hydrolysis, transferring it into a nitrocellulose paper, then we block with excess DNA, which is the probe, and then we wash it off the excess part, and then the autoradiograph, observing it under the autoradiograph. So this is a gist. The same thing is written, that the digestion occurs with a restriction enzyme, and then what we do, we run it on a gel. Then comes the thing of alkali hydrolysis and transferring. So you can see that we have alkali solution over here, and then we have placed a sponge over it. After that we have the agarose gel, and above that we have the nitrocellulose paper, and then in order to stack it we have paper towels or the filter papers. Now the filter is incubated under hybridization conditions with your specific radio label DNA probe, and this probe is complementary to the DNA restriction fragment, and then we wash off the excess probe and then observe it under autoradiogram, and we perform autoradiography, which reveals the DNA fragments to which the probe is hybridized. So what are the different applications of this technique? So we have seen or observed that these kind of techniques can be used in your gene discovery, for gene mapping, then your developmental studies, sometimes your DNA fingerprinting. You can use them in diagnostics, forensics for your DNA fingerprinting, then your preparation of RFLP maps. Okay, and other evolution studies you can also perform. So also it helps to identify the transferred genes in your transgenic individuals. So these are the two important applications of your southern blotting. If we see that it has actually wide applications in different fields, from your gene discovery to a large scale like your forensics, like your DNA fingerprinting and mapping, then evolution, and also identification of your transferred genes. So this southern blotting is what we do for DNA. Now coming to northern blotting, for when we have to detect some kind of specific RNA sequences, so then we also require a northern, what we do. So this is a different name. The technique remains the same, it's blotting, but it's called the northern blotting. So it was developed in the year of 1979 by two professors of Stanford University, and so northern blotting, it was developed by James Alvin and George Stark at the Stanford University, and was named such by analogy to the southern blotting. So what are the steps involved in your northern blotting? So it is that RNA is isolated from several biological samples, like your various tissues, then the different developmental stages of the same tissue, like if we take it in different years. So if we calculate year samples of say a child, and we check as to how the tissues are developing and all, so we isolate the RNA from such biological samples. But we need to also keep in mind that RNA is more susceptible to degradation than your DNA. So then what we do is we load it on a gel, and then we do perform is agarose gel electrophoresis. So these are 32 P labeled sized markers. Okay. So then we separate it based on this size, and this is the resulting gel of what we observe after the electrophoresis run. And then the gel is then blotted on a nylon membrane, or on the same thing, the nitrocellulose filter paper, or sometimes we use is diazobenzyl oxymethyl papers, for creating the sandwich arrangement. So here also we have the gel. So in the bottom we have the salt solution, above that we have the gel. Okay, above the salt solution we have a sponge, above that we have the gel. Then we have the nitrocellulose membrane followed by the paper towel. So the solution passes through the gel and filter to the paper towels. And then we have the gel. Then we get a replica of the gel after the transfer process on the filter, and then so we can tell that RNA has been transferred to the filter. Now the membrane is placed in a dish containing hybridization buffer with a label probe. So we use the label probe in a particular buffer. We call that as the hybridization buffer. Thus it will hybridize to the RNA on the blot that corresponds to the sequence of interest. Now the membrane is washed to remove the unbound probe. So these probes are very specific and will bind to particular sequences only. So then the excess buffer or wash buffer is used to actually remove the unbound probe. So as much amount of probe is there, it will bind, the other things will be removed off. So the probe is hybridized to your complementary sequence. Okay, and we use that in a particular hybridization buffer. And then what we do, the label probe is detected via your autoradiography or via a chemiluminescence reaction. Now if a chemical labeled probe is used, now we observe it under chemiluminescence if we use a chemically labeled probe. Now in both the cases the result is the formation of a dark band on an X-ray film. So that's an autoradiogram. Now the expression patterns of the sequence of interest can be compared, and then you can tell which of your fragments are what. So basically the entire process is same. Like you have salt solution, then so first you isolate it from different biological samples, then you run it on a gel to separate them based on their size, and after that you transfer it on a, so these are the types of, so these are so what filter papers we use, that is important to know as to what is the composition of the filter paper that we are using. It can be nylon membrane or a nitrocellulose membrane or diazobenzyl oxymethyl papers. So sometimes you can be given questions like that, and also you need to keep in mind the arrangement in which they are placed. So you have a salt solution, then you have this then you have the sponge, after that you have the gel, then the nitrocellulose filter, after that the stacking things like you're stacking it with the different filter papers and all, and then you add the probe in the presence of an hybridization buffer, and then you wash it to remove the unbound probes and then observe it under an autoradiogram. Or you can even perform a chemiluminescence reaction. Okay. So these are the different steps of Northern blotting. So first, this is just a gist of it. So first you isolate from several biological samples, then you load it on a gel, and the RNA samples are separated according to their size on an agarose gel. So these are 32P labeled size markers. The gel is then blotted on a nylon membrane or amino benzyl oxymethyl filter paper by creating a sandwich arrangement, and then you place it on a dish containing hybridization buffer with a labeled probe, and then you remove the unbound probe and detect it under an autoradiogram or via chemiluminescence reaction. So in both the cases you will see the formation of a dark band on an X-ray film. So what are the different applications of Northern blotting? So we can study the different things like we can study the RNA degradation. Like if it has started degrading, then we can run it on a gel and then see the different fragments. But we also cannot tell that this is of your particular RNA is degrading. So we need a specific probe for it. So that's why we need to perform this Northern blotting. Then if we have to study your RNA splicing, that your RNAs have spliced, so we can do that. Also studying the RNA half-life is also a very important application of Northern blotting. Then the different other applications is we can check and confirm the transgenic knockout mice studies, then detection of your mRNA transcript sizes, then study of gene expression at the level of messenger RNA transcript. So these are the different applications, and these have other wide applications which you will be finding if you are doing northern blotting, and also different studies can be done based on our RNA experiments. So we can perform the northern blotting, but this northern blotting has several few disadvantages, one of which is that the standard northern blot method is relatively less sensitive. So from the sensitivity point of view, it lacks a few of its features. Then detection with multiple probes becomes a problem. So only when this probe is specific to your particular RNA, then it becomes very important. So detection with multiple probes becomes a problem. Now, even if the RNA samples are slightly degraded by RNases, the quality of the data and the quantification of expression is quite negatively affected. So you will see that even when your RNA is degraded very slightly, like to a very say 10% of it or 5% or 2% of it has degraded, but still you will see that you're not being able to analyze the data or quantify it properly. So this has a negative impact on this kind of blotting techniques. So these are the three disadvantages which northern, okay, so this is not plotting, this is northern blotting. And so this what it has. So the first one being, it from the sensitivity point of view, it is relatively less sensitive, and the using of multiple probe becomes a problem, because you see different sequences of RNA require different probes. So we need to provide a number of probes to actually such that it binds to the different sequences and for the hybridization process. So that becomes a problem. And the third is that the degradation of using some kind of RNases and all, so it will also affect your data and hamper your quantification process. Now coming to the last technique, which is the Western blot technique. So Western blotting was developed in the year of 1981. It's an immunoblotting technique which relies on the specificity of binding between the protein of your interest and a probe. So this probe is an antibody which is raised against that particular protein, and it allows the detection of the protein of interest in a mixture of many other similar molecules. Now the SDS page technique is a prerequisite for Western blotting. Like for the previous steps, we were doing agarose gel electrophoresis and then transferring it. So for this, just because it deals with the proteins and all, so we have to perform SDS page electrophoresis and then we go for the transfer process. Entirely the transfer process is the same, just a few minute changes here and there, and then it becomes same. So here the probe, we need to keep in mind that the probe is an antibody which is raised against that particular protein. So this is important. Now, what are the different steps of Western blotting? So the protein sample is subjected to electrophoresis on an SDS polyacrylamide gel, and then you allow it to migrate. Then electroblotting transfers the separated proteins from the gel to the surface of a nitrocellulose membrane. Okay, and then you transfer it via some kind of electric current. Now the blot is incubated with a generic protein such as your milk proteins or your BSA, which binds to any of the remaining sticky places on the nitrocellulose. Okay, now an antibody that is specific for the protein of our interest, that is the primary antibody, is added to the nitrocellulose sheet and it reacts with the antigen. Now only the band containing the protein of interest binds the antibody, because this is specific, forming a layer of antibody molecule. So as you can see over here, only that band which contains our protein of interest, because we have designed the proteins or the antibody in such a manner that it is going to bind to our only protein of interest, and then it binds over it and forms a layer of antibody molecules. Now what we do is, after washing, then we wash it to remove any other antibodies, and also after washing, for removal of non-specifically bound antibody, that is the primary antibody. A secondary antibody is added which specifically recognizes your Fc domain of the primary antibody and binds it. Now the secondary antibody is radioactively labeled or is covalently linked to a reporter enzyme which allows to visualize the protein antibody 1 antibody 2 complex. So what we see in this diagram is that we have a target protein. So first of all, what we do is we block the other proteins and all. So we incubate it with milk proteins and BSA, which is going to block it. And then we use the primary antibody membrane. So it will bind only to our protein of our interest, and then we provide the secondary antibody, which is going to bind to the Fc region of it. And then we, so this secondary antibody is actually radio labeled, and it is covalently linked to a reporter enzyme. Then we provide a substrate, and after that we get an enzymatic reaction and we can see some kind of color. Then we see under it it under a colorimetric colorimetric channel and all. So here so what we do over here, we prepare the samples, that is, and then we extract the protein. So lysis depends on the tissue. We culture the cells, we sonicate it, tissue samples, then we homogenize. We centrifuge it to remove the debris and keep cold and use the protease inhibitors and the phosphate inhibitors. Now what we have got our protein of our interest, and then we run an SDS page. So this is for the separation of proteins according to their molecular weight, and your proteins are denatured before your SDS page. And finally comes to the protein transfer. So if you can see, you have a cassette where you have the filter pads, then you have the gel, then above that you have the membrane and another set of filter pads. Then you keep it under a say a cassette, and then tightly bind it, tightly screw it, and then the transfer of the separated proteins onto the membrane, which can then be probed with your antibodies to detect the protein of interest. Now the membrane can be of your nitrocellulose or polyvinyl difluoride membranes can also be used. Now we go with the blocking of the membrane. So for that we require a blocking solution which includes your milk or BSA. So fill up the space of the membrane to prevent non-specific antibody binding. So for that we use some blocking buffers, which is your milk or BSA, and then we incubate it with, and then you can incubate it with a primary antibody and then the secondary antibody. And after that so your secondary antibody is tagged with some kind of reporter enzyme, as I was saying, and then you provide a substrate to it which is going to show an enzymatic reaction, and then you will visualize it under, you can do it under an X-ray film development or a luminescence detection or fluorescence detection. So it also has a wide variety of applications. Okay, so one of which is that it is necessary for the confirmatory test of your HIV, and also your Western blot can be used as a definitive test for your bovine spongiform encephalopathy, and some forms of Lyme disease testing also employs this Western blot. So these are the different tests which actually use these Western blot techniques and all. Okay. Okay, so that's all we had with the lecture part. So this is a very short lecture. So let us discuss the questions. So from this lecture, or from this part of the thing, you can expect just direct questions. Nothing would be much complicated and all, or you cannot expect questions which are going to be very tricky. So the questions are going to be very direct and you can answer them very easily. Okay, so I guess I can yes, let me see. Okay, so these are very direct questions. We can wrap up quickly. So let's go with the first question. Right. So Western blotting is done to detect. Yes, specific proteins. So southern blotting for DNA, northern blotting for RNA. Specific protein means your Western blotting. And can you tell me what we do to see specific DNA protein complex? What experiment we do? Yes, we can do that. Also, sometimes we do is IMSA. Okay, that is electrophoretic mobility shift assay. That can also be done. Okay, good. In Western blotting, the first step is to. So, what is the first step of a Western blotting? Run a SDS, run an agarose gel, transfer the proteins in the membrane, add primary antibody to the sample. So you can see that these are very direct questions. There won't be any tricky questions like there won't be equations or your sums and all. Yes, right. 2A is right. It is running off an SDS page. Correct. So question number 3. The proteins of the sample are separated using SDS page, Western blot, protein purification, immunoblot. Yes, so you separate the proteins using SDS page. Yes, okay, that's very good. Okay, the next one is Western blot technique is also known as. What is Western blot technique also known as? Southern blotting, hybridization, immunoblotting, immunoprecipitation. Correct? Immunoblotting is the right answer. So Western blotting technique is also known as immunoblotting. So see, these are very direct questions, nothing tricky. And these are the questions which you are going to get in your exams as well. Okay, the next question. For DNA fingerprinting, which of the following technique is used? So the name says itself. Yes, it's southern blotting. So we perform southern blotting for DNA fingerprinting, and this is what is being used as a wide application of southern blotting. Which of the technique is suitable for detecting a specific mRNA expression in cell? Yes, northern blotting, because northern blotting is used for the RNA techniques. Correct. Okay, the next question is the blotting techniques. A probe is. So, what is a probe in blotting technique? A specific protein to DNA to detect DNA. A short piece of labeled double-stranded DNA which is complementary to your specific DNA. A short piece of labeled single-stranded DNA or RNA which is complementary to your specific DNA or RNA, or all of these. Yes, very good. So it's a single-stranded DNA or an RNA which is complementary as well as specific for that particular DNA. So it is going to get hybridized to that particular, say your DNA which you want to target. It's going to be double-stranded, and then we can visualize it under an autoradiogram. Correct. Okay, the next question. That's question number 8. The non-specific sites on a nitrocellulose membrane can be blocked by. So how can we block the non-specific sites on a nitrocellulose membrane? See, sodium chloride. Are you sure? I was just teaching it. What can we use to block the nitrocellulose membrane? So that's a blocking buffer. We do not use a salt solution for blocking. You are actually misplacing it. The salt solution is used for transferring, where we have the salt solution. So for blocking buffers, what we use in Western blot, such that it blocks. No, we do not use sodium dodecyl sulfate. We use BSA or powdered milk. We use milk or BSA to block it, such that your the all the proteins gets blocked, and the primary antibody is going to only bind to your specific protein of interest, or the protein of interest which you are going to target. So after you have blocked the proteins or incubated it with the blocking buffer, the proteins gets blocked, the layer gets blocked, and then you put it in primary antibody. It's going to bind to your protein of interest, then you provide the secondary antibody, and then the process continues. So it is D, it's rehydrated powdered milk, or sometimes we use BSA. Okay. So this is one question which you can expect, because which is the blocking buffer which we use. Okay, why is the probe labeled? Improve binding capability, improve visibility, improve stability, improve location identification. Yes, it does improve visibility, but a more appropriate answer to this would be improve the location identification. Yes, we use a probe, we know that it's for visualizing only. But see, we have a probe, we want to visualize it, but what we want to visualize? So what we want to actually improve, is it the visual visibility which we want to improve? No, we want to improve the location where these particular things of interest or the molecules of interest are present. So we want to improve the location, such that we want to visualize the location of that. So the location here is important. The visibility is not important. The location or where it is present is important. So yes, it is correct, improve the visibility, but a more appropriate answer to this would be improve the location identification. So question number 9, B is the right option. Or if you are given an MSQ, I think B and C both would be correct. Coming to the next question, what is the major force behind protein and nucleic acids to go out from the gel onto the membrane? Electric current, pH gradient, pressure. There is no option D. You can give temperature also. So what is the force? Is it pressure that we are providing when we are placing it? So the gel is at the bottom, the membrane is in top of the gel. Where is the pressure coming from? So is it because of the pressure that it's getting transferred? No, it is the electric current. We are providing an electric current because of which the transfer is occurring. It is not happening because we have a gel, then we place a membrane on top of it, and this with time it gets transferred. It's not like that, like what we do in the during our, when we were kids, we used to eat some toffees and all, and then we used to get some tattoos, and then we place it, and then we rub it, and then we take it off, and we have the tattoo in our hand. It's not like that, that because of the pressure we are doing it. It's like that we are providing something. So the gel is at the bottom, the membrane is on the top, the pressure factor won't be there. Had it been that the gel is on the top and the filter is, or the membrane is on the bottom, then that situation might have arisen. But as of now, the gel is at the bottom and the membrane is on the top, so we have to go from bottom to the top. So we are providing electric current, because of which the bands are getting transferred, or these particular we are getting replica of our gel image. So it's because of the electric current. Okay, did you get it? It's because of the electric current, not the pressure, because there is no pressure factor which is playing a role over here. Am I clear? Am I audible? Okay, that's good. Okay, now coming to the next question. In northern hybridization, probe hybridization forms RNA antibody hybrid, DNA DNA hybrid, DNA RNA hybrid, all of the above. So what happens in northern blotting, northern hybridization probe? What complex is formed? Yes. So we know that northern blotting is done for an RNA, and then we are providing a DNA, single-stranded DNA, which is going to bind to your RNA, which is going to act as the probe. So 11C is the correct option. Okay, good. Question number 12, to detect protein in western blotting. Antibody is used, antigen is used, DNA probe is used, RNA probe is used. What we use? Antibody. Very good. Okay, nothing to explain over here. Okay, question number 13. If you want to strip an already used membrane for western blotting, which of the following membranes is best to use? Nitrocellulose, PVDF, nylon, all of the above. If you want to strip an already used membrane for western blotting, which of the membrane will you be using? Stripping means you have to, first of all, you have transferred it on a membrane, and then you have to remove it, and then you are using that particular membrane. No, all of the options are not correct. Uh, it is actually PVDF membrane. So this membrane can only has the capacity to be, so can be reused from time to time. So other membranes cannot be reused because they cannot be actually cleaned off and reused again and again. But PVDF is the membrane which we can reuse. So first of all, we have transferred it, and then we want to reuse it. We remove it off, we wash off the entire thing, and then we reuse it. So PVDF, keep this in mind, it is the PVDF membrane. So 13B is the right option. Okay. Question number 14. What will happen if you forget washing the membrane after incubation with primary and secondary antibody? So what will happen if you do not wash it after incubation? Background noise will be very high, whole blot will be dark, no specific band will be observed, all of the above. Whole blot will be dark. So yes, that is true. And so whole blot will be dark means what does it say? Whole blot will be dark. So what does that mean? Your background noise is high. So just because you haven't washed it, the primary antibody is there. Yes. So you have only given the answer. So whole blot will be dark. So then it has the entire primary antibody has bound. The entire secondary antibody is binding to everywhere, and you're seeing everywhere there is band. So the entire blot becomes dark. No specific band you can observe, and finally the background noise is high. So D is the right. All of the above you can see if you forget washing. So keep this in mind. Washing is always very important to remove the additional extra antibodies from your membrane. The last question, why ghost bands appear in blots? Blocking was not done, too much protein has been loaded, incubation with primary antibody was too long, too much substrate for secondary antibody was added. This is an MSQ. Yes, that can be correct. Okay, that blocking was not done, and because of that, you would be observing a few other bands which is not of your band. That is true, but you also need to see that what is the role of this primary antibody, that it is very specific and it is going to bind to your particular protein of interest. Now if you're incubating your say the membrane or your proteins to your primary antibody for a very long period of time, first of all the specificity of the primary antibody gets lost. Then there might be high chances that the primary antibody is binding to your other proteins as well, because you are keeping it. The primary antibody does not want to sit very idly and will bind with the other proteins as well, and then you would be seeing other ghost bands as well. So C is also right. Or sometimes you also see that if you have loaded enough of the enough of your protein. Okay, so if you see that you have loaded enough of your protein, that can be also one of the reason of seeing ghost bands, because see you are doing the transfer process and enough of protein is there, so then there is chances that these bands appear as ghost bands in your transfer membrane. So that can also be a case. Like if you're loading enough of protein, first of all is that you can see a drag, or you can see is that uh, your protein hasn't run properly, okay, or the separation has been done properly. So loading of protein, there should be a particular amount of protein which you should load to see proper. So too much of protein loading can also lead to ghost band. So A, B, C, all three are correct. That blocking, if blocking is not done properly, if protein loading is not okay, or too much of protein loading has been done, or if you are incubating it with a primary antibody for too long, that can also lead to the formation of your ghost bands. Okay, so all the three options are correct. Okay, okay, that's all then from my part. And on Sunday I'm going to take just a very interesting quiz session, hoping to see most of you. So if you have any friends and all, you can also inform them such that they come and attend it, because it's going to be interesting. I'm not going to teach anything. You can even ask your questions and all. I would be free to discuss, but mostly it would be a very interesting quiz session where I would be keeping say 5 questions from each of the 12 lectures. So 60 questions, 60 minutes, and it's going to be fun. Okay, so that's all then from for today. Okay, thank you.